This is Learning Objective 12. In this video, I'm going to describe how E. coli uses the negative and positive controls of the lac operon to economize on RNA and protein synthesis. First, we're going to talk about negative versus positive control. So, what is negative control? There are actually two types of negative gene regulation. There are repressible operons, which are operons where transcription is on, but can be inhibited when a molecule binds allosterically to a regulatory protein. There are also inducible operons. These are operons that are usually off, but can be stimulated or turned on when a specific molecule interacts with a regulatory protein. And this type of negative control is seen in the lac operon. Positive control takes place when an activator protein binds to the DNA. Now we're going to take a look at the role of negative and positive control in the lac operon, and we're going to see how they economize RNA and protein synthesis. First, we're going to look at negative control. The inducer, allolactase, inactivates the lac repressor, which then stimulates the synthesis of enzyme beta-galactosidase. This, in turn, catalyzes the breakdown of lactose. This is economization, because enzymes to break down lactose are only synthesized in the presence of lactose. This means unnecessary production of the enzyme doesn't occur. The cell doesn't waste energy or time producing things that it doesn't need, in this case, an enzyme to break down lactose. Here I have some really great diagrams that are going to show us the role of the negative control in the lac operon. So first, we're going to take a look at our lac operon when lactose is absent. So, as you can see here, we do have an active repressor. What's unique about the lac operon is that its lac repressor, aka active repressor, is actually able to attach to the operon on its own. So here you can see that our active repressor is able to attach to our operator, preventing the production of beta-galactosidase. Now, we're going to take a look at what happens when lactose actually is present. So, here you can see that we've got what's called an inducer. This is going to bind to our protein, aka our active repressor, and it's going to prevent it from turning off our lac operon. So, you can see then that RNA polymerase is able to bind to the operon, allowing for the production of the enzymes which break down the lactose. Now we're going to take a look at the role of positive control in the lac operon, and we're going to see how it economizes RNA and protein synthesis. So positive control really takes place when there is very little glucose within the cell. There's a scarcity. Cyclic AMP will accumulate within the cell due to this lack of glucose. The cyclic AMP is going to bind to catabolite activator protein, also known as CAP. This binding between the two will cause the cap to assume its active shape. When it assumes the active shape, it will bind to the lac promoter on the DNA, increasing the affinity of RNA polymerase for the promoter, causing an increase in the rate at which the lac operon is transcribed. This can be seen as economization of protein synthesis, because really, if glucose is present within the cell is going to want to use that glucose for glycolysis. It is much more efficient than going about creating enzymes and creating sugars out of lactose. But if there is no glucose present, then the cell will need a means of survival. It's going to be forced to create enzymes to break down that lactose into simple sugars to create ATP. So, if there is glucose present within the cell isn't going to waste time going through the processes of creating enzymes to break down lactose into simple sugars, when it can use glucose to create the ATP it needs through glycolysis. So I know that was all a lot of information to digest. So we're going to take a quick look at these diagrams to help better our understanding. So looking up here at our first diagram, we're going to see what happens when lactose is present but glucose is scarce within the cell. So, in the event that there is a scarcity of glucose within the cell, we are actually going to have an accumulation of cyclic AMP. This cyclic AMP is then going to bind to our cap, which causes the cap to take on its active form, its active shape. When it takes on the active shape, it's going to bind onto the lac promoter on the DNA. This increases the affinity of RNA polymerase for the promoter, causing it to bind. So, if glucose is scarce, the high level of CAMP activates CAP, and the lac operon will then produce large amounts of mRNA coding for the enzymes in the lactose pathway. Now, we're going to take a look at what happens when both lactose and glucose are present. This is fairly simple. If glucose is present, then we're going to have very low levels of CAMP. Because when glucose is present, CAMP is scarce, and CAMP cannot bind to inactive CAP, we cannot stimulate transcription. So, I'm going to repeat. If we have glucose present, we have no accumulation of CAMP, which means we cannot bind to CAP, and we won't be producing the enzymes needed to break down lactose. Now this can be seen as economization because when glucose is present, even if lactose is also available, the cell is going to preferentially catabolize glucose. So it's going to make really little use of the lactose-utilizing enzymes. It's not going to waste time and energy creating enzymes needed to break down lactose into sugars when it's already got the sugar glucose available to it. And that concludes our video.